Hello everyone, welcome to Mbbs Hub. In our today's topic, we are going to learn about the molecular cell biology in health and aging. Okay, coming to the plasma membrane. Plasma membranes mean what? Membrane lipids and glycolipids, and it is a major component of the cell membrane is lipid. Lipid is the major component of a cell membrane, and it occupies and it occupies 20 to 80% depending on the cell. Okay, coming to the phospholipids. Phospholipids are main lipid constituents, okay, of biomembranes. It is bilayer. Okay, in this we will see hydrophilic and hydrophobic. Hydrophilic are the aqueous environment, and which they can absorb water. Okay, the polar heads are in two layers like this. Okay, hydrophobic, these are repelled by water. They cannot absorb water, or they will be no wet on that layer. Okay, those are hydrophobic. And then cholesterol is the molecular of cholesterol inserted between the phospholipids. Okay, molecules of cholesterol inserted between the phospholipids prevent tight packing. Cholesterol prevents the tight packing of phospholipids molecules. Okay, then glycolipids. The endoplasmic surface of a cell membrane. Glycolipids are on endoplasmic surface of cell membrane. Carbohydrate sugar chain attached to the lipids. Okay, these are mainly seen in the myelin sheath. Then the functions of plasma membrane. It protects the integrity of interior of cell, and it prevents unwanted material out of the cell, and that means the unwanted material will be in the outside of the cell. They will not enter into the cell. Okay, then transport nutrients into the cell. Nutrients will be entered into the cell. Okay, and metabolic waste outside. Nutrients, metabolic waste. Okay, they prevent loss of needed metabolites. They will be less secretion, or they will help for keeping the metabolites in the cell. And then they maintain ionic composition, pH, and osmotic pressure in the cytosol. They provides anchor to cytoskeleton and provide that supports cells and helps it maintains its shape. Then coming to the extracellular matrix. Extracellular matrix is a collection of network of extracellular molecule secreted by distinct supportive cells. This is not that much important, but I am telling. Like, okay, now the extracellular matrix functions are structure supports to cell stiffness and elasticity, segregates tissue from one another, regulate growth and differentiate of cells by growth factors, essential for wound healing and fibrosis. Okay, extracellular matrix. These are the main functions of the extracellular matrix. It is composed of fibrostructure proteins, gel-like fluids, and other glycoproteins. Okay, coming to the cytoskeleton. There are three types: microtubules, microfilaments, and intermediate filaments. These functions are shape and support to cell interrelation with other intracellular organelles, mobility, communication. Okay, then mitochondria. It is a double membrane. Outer membrane has proteins and passage of ions. small molecule of proteins, and ATP generation, active sites for ATP generation, conserved genes for tRNA, rRNA genes of ion transportation and ATP synthesis. Its functions are ATP synthesis and energy generation by ATP synthesis, mitochondrial DNA, and independent multiplication. Endoplasmic reticulum. There is a rough endoplasmic reticulum, smooth endoplasmic reticulum. Okay, they are interconnected with Golgi and plasma membrane for transport by vesicles. Rough endoplasmic reticulum is for protein synthesis and synthesis of antibodies. Insulin, erythropoietin, etc. And smooth endoplasmic reticulum is for synthesis of membrane lipids, carbohydrates. Okay, these are a transitional zone for transport of RER products. Okay, we will all learn this in our intermediate. And then ribosomes, they contain rRNA, ribosomal RNA. Okay, and other proteins. New protein synthesis by transcription from DNA to mRNA, followed by translation by rRNA. Bound from RER, main synthesis for protein, synthesis of new protein. Then Golgi apparatus, there is not that much required. Then this is the structure of the cell. It is a microvilli, microfilaments, janula adherents, janula, these are adherents, microtubule, lysosome, Golgi apparatus. Okay, these are microtubules, these are intermediate filaments. And these are microfilaments, microtubules, microfilaments. Okay, these are intermediate filaments. Functions are structure and support, intercellular transport, contractility, motility, and interrelationship with the other organelles, and intercellular communications. These are the functions of this filaments. And coming to the cell cycle and its regulators. Cell cycle, it's a study of coordinated series of events lead to cell replication. Okay, cell cycle, and it is a study of coordinated series of cell replication and successive divisions of a cell that is connected into a daughter cell. Okay, that is a cell cycle. In this there are two phases, interphase and mitosis. Okay, we generally know that interphase is more than the mitosis. Okay, coming to the interphase. In interphase there are three phases, G1, S, G2, gap 1 phase, synthesis phase, and gap 2 phase. Okay, in gap 1 phase the cell prepares for the DNA replication. Okay, the cell prepares for the DNA replication, and cell will grow by the building up proteins and organelles. Okay, this is the longest phase. One phase is longest phase, and it has a checkpoint. Okay, for to check the readiness of the DNA replication. Here we can see it is a G1S checkpoint. Okay, I will show you here. See, this is G1S checkpoint. This is G1 phase, and this is synthesis phase. In this there is a G1S checkpoint. DNA replication will happens. Okay, will continues in this readiness of the DNA replication. Okay, In this S phase, it is a synthesis phase. Cell growth will be continued in this phase, and the replication of the chromosome is occurs in the cell. Okay, it contains a double number of chromosomes, and S phase contains 2 chromosomes, double chromosome. Okay, next, gap 2 phase. It is the cell continued growth will be there, and it prepare for the mitosis. In this also there is a checkpoint. It is a G2M checkpoint for checking of accurate replication of chromosome. Okay, I will show here. This is G2M checkpoint, accurate replication of the chromosome. Okay, then coming to the mitosis. In mitosis there are 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Coming to the prophase, it is a chromatin condensation will starts here, and then nucleolus will break down and disappears. Okay, in the late prophase, the kinetochores are formed. Okay, in the prometaphase, the nuclear envelope disintegrated into small membrane. Okay, the kinetochore microtubule will connect to chromosomal and form mitotic spindle. Okay, this next, metaphase. There are paired chromosomes split up at centromeres. And form a moves to opposite sides. Okay, and after that they will form a equatorial plane. This is called as the equatorial plane, and then equal distribution of the chromosomes. In this, the chromosomes will be equally distributed in the metaphase. We will see M checkpoint. I will show you. This is M checkpoint here. Okay, this is M checkpoint. After that, it will go into the, it is directly shown, there is a G0 phase. Then after that, anaphase. It is a newly formed identical doctors can be seen, chromosomes, daughter chromosomes, not it moved to the opposite ends and condensed and ready to form new nucleus, and at the end new nucleus is formed. Okay, then final step, it is a telophase. Elongated, and the cell is elongated, and polar microtubules, and the both daughter chromosomes are segregated. Okay, the nucleus also reappears, and after that, last resting phase, G0 phase may or may not be, will not happen. It will, if in the condition, it will re-enters the mitosis. Okay, after that, cellular basis of aging. In cellular basis of aging, cell slowly declines to function. Okay, and structural functional changes in different organisms and organelles and system of human body with aging. It will also decreases, and there are some theories of cellular aging. In that, first one, telomere shortening and cellular senescence, free radical theory of aging, mitochondrial dysfunction, DNA damage, altered gene expression, reduced autophagy, activation of tumor suppressor genes, and caloric regulation theory. Coming to the telomeric shortening and cellular senescence. In this this theory is about the telomerase activity. Generally, the telomerase activity is for the repairing the telomeres and activity, so they can more cells will be created, renew cells. Okay, in somatic cells there is no telomerase activity. Okay, this exists in the telomerase activity exists in the germ cells. Okay, that means telomerase activity means the repairing of loss of telomere. Okay, the somatic cells has no telomerase activity. This causes the inability to renew cells. There will be no new cells. Okay, and third one, mitochondrial dysfunction. Mitochondria generally, the mitochondrial dysfunction will leads to the ATP declination, decrease, and then oxygen free radical increases, causes mitochondria and vacuolization. Okay, and after that, DNA damage. In DNA damage there are two phases, exogenous and endogenous. In exogenous properties there are physical, chemical and biological, and in endogenous properties there is reactive oxygen species. In this, Werner syndrome is there. It is a case of defective DNA helicase. That means it is also called as a premature aging, which is a defect in DNA repair enzymes. Having a DNA repair defect in a DNA repair enzymes is called as a premature aging. Okay, premature aging or Werner syndrome, which is a defective DNA helicase. Werner syndrome. And then the fifth one, altered gene expression. In altered gene expression, reduced expression of many genes and proteins associated with mitochondrial function. The decrease in the expression of many genes. Okay, the gene expression will be reduced, and protein associated with the mitochondrial function is also decreased. Sixth one, it is a reduced autophagy. I mean, the theory, reduced autophagy. The sixth theory, it is a accumulation of waste products. Reduced autophagy means it is a accumulation of waste products. And then seventh theory, it is activation of tumor suppressor genes. In this, regulate the cell division and implicate both cancer and senescence. In this, both arrest of cells will be done by the apoptosis or senescence. In apoptosis means it is a cell suicide. Apoptosis means cell suicide without there is no inflammation. Inflammation will not be there. Okay, senescence means what? It is a stoppage of cell division. Senescence. Okay, stoppage of cell divisions. And the last theory, it is a caloric regulation theory. In this, caloric restriction reduces the release of growth hormones, insulin growth factors, and insulin growth factors like IGF-1. Okay, this delays aging and these can be cause for risk for diabetes, hypertension, cardiovascular diseases, and cancer. In extreme obesity conditions, there is a high risk of dying early. Okay, there are high chances for dying early in extreme obesity. Coming to the diseases according to the cellular aging, first one, cardiovascular system. In we'll see about atherosclerosis, arteriosclerosis with calcification, and Monkeyberg's medical calcification. This is also important slide. We should know about it. In this tunica media, is enlarged. Tunica media is enlarged in this condition. Enlarged. And I mean, tunica media means if a blood vessel, a blood vessel will have three layers: tunica adventitia, tunica media, and tunica externa. Okay, in that tunica media is enlarged, and the nervous system diseases like atrophy of Gary and Salke, Alzheimer's, Parkinson's disease. For Parkinson's, we will use L-DOPA. Okay, it crosses the blood-brain barrier. These are the minimum we will remember it. And the musculoskeletal system, degenerative bone diseases, loss of bone density, age-related muscular degeneration, and for the eyes, deterioration of vision due to cataract and vascular changes in retina, and hearing disability, and in immune response system, reduced RGG, immunoglobulin G, and response to antigens, and in skin, lack of elastic tissue. The skin will be అంటే ముడుచుకుపోవడం అని అంటారు కదా స్కిన్ అనేది, సో దాన్ని లాక్సిటీ అని అంటారు అనమాట. ఫోల్డెడ్ skin folding of the skin we will see in the old persons. Okay, and then cancers occur at the age of 50 to 80 years. So this is our today's topic, guys, and thank you for watching. We will meet you in the next lecture.